Hello, my name is Kyle, and in this presentation, I will be talking about the regulation of sperm maturation in the germinal epithelium. So the top image showcases the male reproductive system, and we can think of the male reproductive system as a series of ducts and tubules that serve to transport mature sperm cells to a destination outside of the body. The bottom image shows the testes, a closer look at the testes, and it is this internal genitalia that we want to focus most of our time on because it is the primary site for sperm maturation and sperm production. Also highlighted are the epididymis. These are structures that also could play a role in sperm maturation during the final stages of the development, but we will go into that more in detail later in this lecture. For now, by the end of this lecture, there are 3 learning objectives that we want to achieve. The first is that we want to understand the stages of sperm development from spermatozoa to spermatogonia to spermatozoa. The second, we want to gain a better understanding of the components of the testes that influence sperm maturation. And finally, we will examine the hormones that play a key role in spermatogenesis. Okay, so in this slide, I wanted to focus on sperm development. And at birth, the testes of a newborn male have not progressed beyond mitosis. So we're right here. And the male gametes are inactive until puberty. Now, puberty hits, and the testosterone synthesis has increased drastically. And spermatogenesis will kick into high gear. And at the start of spermatogenesis, the male germ cells, these now active male germ cells, are known as spermatogonia. So we're right here, and we are also up here. Now recall that in order for sperm to combine with a female gamete, each of these germ cells, they have to be haploid and must contain only one copy of that chromosome. To create a haploid sperm cell, the spermatogonia has to undergo 2 meiotic divisions, so 1 and 2, to transform from 1 parent cell into 4 haploid daughter cells, which you can see why there is a lot of sperm development as the male ages. All right. Also, throughout the process of spermatogenesis, the developing sperm cells move from the basal compartment of the germinal epithelium to the tubule lumen. And this is and the sperm, so the sperm cells will mature from the spermatogonia to the primary spermatocyte, to the secondary spermatocyte, to spermatids, and then finally to its mature form, spermatozoa. The spermatozoa, maturation of the spermatozoa is crucial because it develops this tail. And from the spermatid to the spermatozoa, we can see in this far left image that the Golgi complex will transform into this acrosomal area. The cytoplasm of the spermatid is mostly lost in this final stage, mature sperm cell. The nucleus will make up the head, the centriole and the mitochondria make up the body, and the microtubules will transform into the tail or the flagellum. Motility of mature sperm cells outside of the epididymis to the vas deferens and so on is beyond the scope of this presentation, but it is important to mention. All right, so by understanding the stages of maturation, we can now focus on the testes, which serve as the primary site for sperm production. So the cutaway image here on the left shows that the testes are composed of a lot of coiled tubules. These are the squiggly lines right in the middle. These are known as seminiferous tubules. And if we magnify this image further, we notice that the seminiferous tubule has two types of important cells, which are highlighted in yellow. The sperm cells, all forms of development, are the ones that are in blue. And these, as you can see, are stacked from spermatogonia and all the way up into, as they develop into the spermatozoa. You'll also notice Sertoli cells. And Sertoli cells is this purple large cell right here. And these are, they extend from the bottom of the basal lamina all the way to the tubular lumen. The other important cell type highlighted in green is the interstitial cell. And they are located between, in the interstitial space between other seminiferous tubules. Lastly, I want to mention that sperm maturation can be completed in the epididymis through the bulk flow. So bulk flow of immature spermatozoa will push different types of sperm cells at different developmental levels toward the lumen and into the epididymis. And the sperm will then complete its maturation stage in the epididymis prior to being ejaculated from the body. Okay, in this slide I wanted to take a closer look at two key cells, the Sertoli cells and the Leydig cells. First, the Sertoli cells link together in the seminiferous tubule to form tight junctions, and these tight junctions form the blood-testes barrier, and it serves an important role in forming three distinct compartments. One, the lumen, tubular lumen. Two, the basal compartment of the basal lateral side of the Sertoli cells, so all of this in purple. And then three, the last compartment is the interstitial fluid outside of the basal lamina. Sertoli cells also provide the developing sperm cells nourishment, and it also secretes important cell byproducts such as proteins, enzymes, and hormones. Inhibin. The hormone that's important is the androgen binding protein. The other important cell are Leydig cells, which are down here. And Leydig cells are important because they secrete testosterone. Without testosterone, we would not be able to develop male secondary sex characteristics, or it also plays a key role in male reproductive tissues, testosterone. Okay, now in this slide, I wanted to focus on the hormonal control of sperm development. Now, this control follows a general pathway. So the hypothalamus will secrete gonadotropin releasing hormone, and this hormone, gonadotropin releasing hormone, will promote the release of luteinizing hormone and follicle stimulating hormone from the anterior pituitary. The follicle stimulating hormone and the luteinizing hormone will act on the testes, and the follicle stimulating hormone will stimulate Sertoli cells. Sertoli cells will then release cell products such as the nourishment, inhibin, androgen binding protein, and other paracrine molecules are then produced. Luteinizing hormones will target interstitial cells here, and the interstitial cells will then promote testosterone synthesis. What's important to note here is that testosterone is a small enough hormone that it can cross its lipophilic nature and its small nature can cross these different compartments into the tubule lumen. But because of the Sertoli byproduct of androgen binding protein, the testosterone and the androgen binding protein will link together, and that prevents further testosterone diffusion throughout the three compartments. It's also worthwhile to mention that researchers are still trying to understand the pathway and the mechanism for the role of testosterone and follicle stimulating hormone in the process of spermatogenesis. So lots more research to be had there. Okay, so putting it all together, the takeaways from this lecture, I want us to know that spermatogonia differentiates into spermatozoa in the seminiferous tubules of the testes. The developing immature sperm cells move from the basolateral end all the way to the tubular end of the lumen of the germinal epithelia. And number 3, adjacent Sertoli cells form tight junctions, which then form the blood-testes barrier to control the diffusion of large molecules. And finally, which prevent large molecules from diffusing across the compartments. And then finally, sperm maturation would not be possible without gonadotropins. So without gonadotropins, the Sertoli lydase cells and testosterone would be difficult to synthesize. Okay, that is it. Thank you for listening. All right, that concludes my presentation.